So first, I would like to thank Beatrice for inviting me to do this today. I wasn't really expecting to be in a sound studio. I was expecting sort of a regular classroom, but that's all right. And I also wasn't expecting the lights, or I would have wore a hat. But we'll just work with it. Sunglasses are they're not interchangeable with hats, actually. So the first thing I would say is, a few years ago, I was back home in Houston, Texas with my high school buddies, and one of them was like, man, I looked you up on Rate My Professor. It's like, boy, do they hate you. so yes, understood. So I understand that many of you are students in BSc 2010. It's also great to see Matt Kerrigan again. I've known him for quite a while. I haven't seen him in a long time. And maybe some of you haven't even had introductory biology. Everybody's had high school biology. Okay, so I thought I would start with a little refresher. So you guys are now, the ones who are now in BSc 2010, you have segued into the evolution section, right? Mm-hmm. Okay, so if I start talking about evolution, you guys know what's going on. So if I write this out, let's say, right, that is, you guys recognize that? Okay. And this is basically the Hardy-Weinberg equilibrium formula, right? So there are, how many things will perturb, how many evolutionary forces will perturb Hardy-Weinberg equilibrium? No need to shout. Okay, I could pick people. Everybody, you guys say, I said, okay, you guys recognize this, and you nodded your head. You recognize Hardy-Weinberg equilibrium, one locus, two alleles, right? So she nodded her head. Okay, so if if some things are true, then Hardy-Weinberg equilibrium is true, right? The genotype frequencies remain constant for all eternity, right? So, what are those 5 evolutionary factors that can perturb Hardy-Weinberg equilibrium? So, HWE is true when, and basically you got 5 things that start with no. Yes, ma'am.
No gene flow.
No gene flow. Okay, so we will call that migration, right? So no migration. Okay, migration is an evolutionary force, right? Gene flow. You carry your genes with you, right? So if you move to Kazakhstan and meet a nice, handsome Kazakhstani guy and get married and have offspring, you have brought all your genes from genes
Australia instead.
Wherever. So for reasons lost to memory, Kazakhstan is always my joke population for one of the next speech. So what else? No migration. Yeah. So we got one big population, right? So what else? Jason, what's your name? Tell me your name again. Jared. I knew there was a J in there. No migration. Talking about evolution here. What else?
No genetic drift.
No genetic drift, right? So, so no migration. I'll just write this like no migration. What that means is infinite population size, right? So genetic drift is just the random sampling of alleles in gametes, right? So no genetic drift, infinitely large population. That doesn't sound very realistic. What else?
You can see.
So who's making eye contact here? So you have been making, yeah, you in the green jacket.
Natural selection.
Okay. Yeah, it's evolution, natural selection. So no selection zone. So we got migration, we got finite population size, we got no selection. 2 more. Yes, ma'am, in the back.
I'm thinking no, like, huge, like, ecological factors like hurricanes or continental drift or something.
So hurricanes would likely come into selection,
Okay.
and continental drift would likely come under migration, I would guess, but also genetic drift. So what is the source of the ultimate source of genetic variation?
Go ahead.
I'm sorry. Yes, ma'am.
No mutation.
Right. I don't have that. No mutation. And there's one more. Think your mail order date from Kazakhstan.
Do you, how do you choose a partner? Do you choose a partner?
If you were oysters, so the oysters are my class. How do oysters mate? Oysters are out there on the oyster bar, and they spew gametes out into the water, and the waves swish them back and forth. Random mating. So we assume random mating. MTG, mating. So anyway, I just wanted to get everybody kind of on the same, just warmed up for thinking about evolution and population genetics, which is what I do. So now, I guess the 4-word description of what we do in the Baer lab is understanding variation in genetic variation, right? So if we look at humans, let us paint with really broad strokes. If we look at humans from Africa and humans from Europe and humans from East Asia, the Africans will be more genetically variable than the others, and the Asians will be a little bit more genetically variable than the Europeans, but not very much. And we think we know why that is. It's because humans had their origin in Africa and have been there longer and have had more time to accumulate genetic variation. And then with migration to points north and east and west, some of that genetic variation was lost, so there's or didn't come along, I guess I would say. So we think that's why Africans are more genetically variable than other humans. Same thing is true in Drosophila melanogaster, fruit flies. Drosophila melanogaster from Africa are more variable than Drosophila melanogaster from other places. We think that's because they came with humans. We took bananas with us, and the flies came along on the bananas. So that's the kind of thing I'm interested in. Is when there is variation in genetic variation among groups, and there's all sorts of different things that could be a group. That's what we're interested in. I don't study 2-headed turtles. And quite honestly, I don't know if that is actually a real turtle or whether that is a photoshopped mutation, as it were. Now, occasionally 2-headed vertebrates do turn up, but not very often. There is not much genetic variation out there for 2-headed vertebrates. A good, by way of contrast, somewhere between 2 and 5% of humans have a non-standard number of vertebrae. Is there anybody out there in the crowd? If I get 40 people, I would expect maybe one person to have a non-standard number of vertebrae. And I don't, if you're not comfortable in saying, yeah, I get an extra vertebra, don't feel bad. But on average, I would expect one person out here maybe would have, somewhere between 2 and 5%. My postdoc advisor, Mike Lynch, has an extra bone in his neck. And he told us this at lab meeting one day. He says, yeah, I got an extra bone in my neck. And he didn't discover this till he was, into adulthood. And he says, yeah, well, that explains why when I was a kid and I went to the doctor, the doctor was always like feeling on my neck. I could never figure out. He said, usually that's an embryonic lethal mutation. He said, but somehow he made it through that selective sieve, and he went on to be a famous and important evolutionary biologist. So, another anecdote. So early on in my teaching career, I used the example of polydactyly, which is sort of having an extra digit, because most deleterious mutations that are at least out there in the population are recessive. You guys know dominant and recessive, right? For kind of obvious reasons. But polydactyly, it turns out, or at least some forms of polydactyly are dominant mutations, and the frequency in humans is about 1 in 300. So I was teaching BSc 2010 at the University of Florida, like 300 people in the room, and I said, I'm talking about deleterious mutations, and okay, we got polydactyly, and I showed a slide of some guy with, extra fused fingers or something like that. And I say, it's dominant mutation, the frequency is 1 in 300. So we expect one person in this room on average to be polydactylous. Is anybody out there polydactylous? And this one guy raises his hand, and there were 600 eyes in the room going, and they're like, oh my God, I can't believe I asked that. So, polydactyly is less common than extravertebrae, which is much, much more common than extra heads. All right, so now launching into the talk. So one of the things Beatrice asked me to do was to sort of, give a little bit of context for how I came to be an evolutionary biologist, and we can talk more about that later. But this is sort of my canonical slide. The Baer research program at age 9, it's like, I was the kid who's always like looking under rocks for salamanders. The class goes out on the field trip or whatever, and there's one kid who's sitting there looking at, that was me, right? Love looking under rocks for salamanders, and I have segued into a fun, if not necessarily lucrative career. It's pretty lucrative. More so than what I used to do. Anyway, now what I do is I count worms, which is as glamorous as it sounds. And that is what Olivia Smith, who was in Beatrice's program last year or 2 years ago, and she's working in my lab now, that's what Olivia does, is she counts worms. I also have an image, it didn't load properly, but we can also engineer these worms to be fluorescent, to carry a green fluorescent protein gene, which now everybody knows about because you go to PetSmart, and they got the glowfish, which are just zebrafish that they've put a fluorescent, you get green glowfish, and then we got red and yellow and all different colors, right? So they got all different colors of fluorescent protein. Well, we do it with the worms, and it's really handy because we just turn on the appropriate wavelength light, and they glow green, which is great, but the image didn't load properly. All right, so now segueing from one locus and 2 alleles, which is the very foundations of population genetics. And so if your family is like parts of my family, and at the family reunion you don't necessarily want to tell them you're an evolutionary biologist, I can say, oh, I do population genetics and phylogenetics. And they're like, oh, that sounds really interesting, without it being fraught with baggage. So that's all of evolutionary biology, is either population genetics or phylogenetics. So, I'm talking about genetic variation. So let me just give this example. So here we have an example of 20 nucleotides of 3 DNA sequences, 20 positions. You can count them up, there's 20, right? So, and I've used, for example, this is me. And for example, that is a sequence of my brother. And for another example, that is a sequence of my cousin Jimmy. So we can count up, so if you look, so you can go down from position 1 to position 20. Position 1, that is what we call fixed. There is no genetic variation, at least in this sample, position 1, or everybody's got an A. Same thing, position 2, everybody's got an A. Position 3, everybody's got a G. Now if we get to position 4, me and my brother have an A, but our cousin Jimmy has, me and my brother have a G, but cousin Jimmy has an A, right? So that site is variable. It's what we call a segregating site. Segregating, just remember Mendelian segregation. If you're heterozygote, half the gametes get one allele, half the gametes get the other. Segregating site is just sort of jargon we use. And you can go down the sequence, and the variable sites are colored in red. So what we can see out of this sample is 5 out of the 20 sites are segregating sites, are variable. And then we can calculate, we want to say, well, how different are these, or how similar are these? And we can calculate the average number of pairwise distances. And a pairwise distance is just or pairwise difference is just the number of differences between any pair of sequences, right? So, between me and my brother, there's 3 differences. So we're adding them up to calculate the average. So there's 3 between me and my brother. There's 4 between me and cousin Jimmy. And there's 5 between my brother and cousin Jimmy. So we add them up and calculate the average. So on average, 4 out of the 20 sites are segregating. So 20% of the sites are variable. And so the average number of pairwise distances is, we just call this pi. So what? So this is what I call genetic variation 101, right? Theta equals 4NE mu. And any population geneticist can recite that like a mantra. And in fact, I heard a story about a guy, and I can't swear, I have not seen this personally, but the story is this guy who was a Ph.D. student at McMaster University in Hamilton, Ontario. During his Ph.D. qualifying exams, bollocksed that equation and was so chagrined that he went out and had theta equals 4NE mu tattooed on his bicep. So now like I say, that's an apocryphal story. I have no idea if it's true, but it might well have been true. So this E, that's an expectation. All that means is the average. So the average pairwise distance is, we population geneticists call that theta hat, and that's a hat over it. And that equals a very cosmic quantity in evolutionary biology, which is 4. If it's diploids, it's 4. It's 2 in haploid. 4 times NE times mu, 4NE mu, so theta is the measure of nucleotide diversity, how much genetic variation there is. So the expected average pairwise distance. The expected pairwise distance is very small. There's very little genetic diversity. If it's very large, there's a lot of genetic diversity. So actually I'll start back to front. Mu, that's the mutation rate, the per nucleotide mutation rate. So we got something like 2 times 10 to the 9th nucleotides in our genome. The per generation mutation rate is something like 10 to the minus 8th. So figure out, you can do the math, say how many mutations will our offspring carry that we, new alleles will our offspring have that we don't have, right? And you can do that calculation. So mu is the per nucleotide, per generation mutation rate. NE is basically everything else. So this is the genetic effective, what's called the effective population size. So the population size, the census population size, just how many individuals are out there, right? Humans, it's 6.5 billion or something and counting. The effective size is basically how large, genetically speaking, how large does that population act? And so one key thing is the larger the population, the more genetic variation it can have and usually will have. So if the population size is one diploid individual, let's think about one genetic locus in one diploid individual. How many possible alleles are there at that locus? Let's say, 2, right? You can either be A1A1 or A1A2 or A2A, right? So there's, the most, for any particular nucleotide site, there's only 2 possible, you can't have more than 2 because you only got 2 copies of the gene, right? So if there's 2 people in the population, what is the maximum number of possible alleles?
4.
It's 4, right? Because each of you could have, up to 2 alleles, and those 2 alleles could be different between the 2 of you, right? So you see, so the larger the population, the more opportunity there is for genetic variation. So, larger population, more genetic variation. That's kind of like, that's dog bites man, right, in journalism, this, duh. So it turns out, so if groups, and here's where I'm coming back to, if populations within a species or different species or different chromosomes within a population, for example, consistently have different levels of genetic variation, right? Either the mutation rate can be different, which it may be, or something else has to be different. And that's why I say NE is everything else. And so from my perspective, mutation rate it's not easy to measure, but it is straightforward to measure. It's extremely labor intensive and expensive, but it is straightforward to measure in principle. Whereas all these other things are not nearly so straightforward to measure in principle. So if you want to, if we're evolutionary biologists, we're interested in all these, we're interested in selection and migration and random genetic drift and all these things. Of these things, mutation is by far the easiest, is the most straightforward to measure. So that's where I sort of come in here. If we want to understand evolution, let's build a foundation of understanding mutation. And then we can, once we know that, then we can build on it, right? So here's a classic question in evolutionary biology. So if I was to call you a mutant, would you take that as a compliment or a slur?
Probably a compliment.
Probably a compliment. Oh, well, you're a mutant. Okay. See?
For something different.
Yeah, okay. So, if one of your buddies calls, yeah, man, he's a mutant, right? That's affectionate. If somebody you don't know calls you a mutant, you have to, at the very best, you have to think hard about it, right? So the point being here is most mutations are bad. We think, like genetic disease in humans are caused by mutations that have not, if like, think of that two-headed turtle, having two heads are rarely better than one, evolutionarily speaking. It makes the decision-making process more complicated. But so this is a classic question: is why is the mutation rate not 0? Mutations are bad, and this follows from a basic principle of engineering and life and everything else, which is there's a lot more ways to screw something up than there is to make it better. And you've probably encountered this many times in real life. So you got something that works pretty well, like an organism that has survived to reproduce, right? That's it's worked well enough to live to at least have the opportunity to reproduce. Ideally, it would if you're going to randomly change some of the genes in that organism, the chances are a lot better you'll screw them up and make them worse than you'll make them better. Therefore, it kind of stands to reason that reducing the mutation rate really, really low would be a good thing, right? Okay, you're sitting there like you don't entirely buy that, but okay. So, like I said, this is a classic argument in evolutionary biology, and there's two, up until recently, there were sort of two possible explanations. The first is due to Sir Ronald Aylmer Fisher, who was the second greatest evolutionary biologist after Charles Darwin. And I came to that conclusion a few years ago. Fisher really was the second greatest evolutionary biologist. And I always call him Sir Ronald because when my daughter was little, there was a book called The Paper Bag Princess. Does anybody remember the Paper Bag Princess? Either you were a little girl or you have a daughter who was a little girl. But anyway, there was a scoundrelly character named Sir Ronald in The Paper Bag Princess. And Sir Ronald Aylmer Fisher said in 1930, once you have fixed the most fit allele at every locus, right? So so P goes to 1, the A1 allele is fixed, or the B1 or C1 or D or whatever, right? Once you have fixed the most fit allele at every locus, right? Once you've fixed any allele at every locus, without mutation, further evolution is impossible, right? Stands to reason. I mean, there's no genetic variation, so evolution is not possible. So he's, Sir Ronald specifically said, adaptive evolution. That means, so if the environment changes, if you have a hurricane or something, right, or continental drift, the environment changes, some context of the organism's environment changes, natural selection changes. Without any genetic variation, without mutation, there can be no genetic variation. Sir Ronald said, adaptation is ubiquitous. We see it all the time, every day. Ergo, the optimum mutation rate is not 0. Simple as that. And he's one of the greatest mathematicians maybe ever. No math necessary, just logic. Says adaptation is ubiquitous. Without mutation, you have no adaptation. Therefore, the optimum mutation rate can't be 0. Okay, so that is a little bit facile and unsatisfying. So shortly thereafter, Alfred Sturtevant, who you may have maybe heard from the genetics section of introductory biology, sort of one of the great early geneticists, came up with the idea of, or actually, wait, no. Sturtevant didn't mention the cost of fertility. Mutu Kimura, who was a great population geneticist in 1967, put forth this idea is, so natural selection probably really does want the mutation rate to be 0, or as low as it can possibly get. But, and again, invoking engineering principles, if you make the tolerances finer and finer, it's going to cost more and more, right? A Mercedes-Benz costs more than whatever that brand of Indian car is, or the Russian car, they were sort of cheap, and the tolerances were not so, or Ford pickups from the 70s, which are my personal favorite example of loose tolerances. You want to increase, so if you want to make DNA replication better, more accurate, it's going to cost you, both in terms of the cellular resources you have to devote to DNA repair, and there's lots and lots of DNA repair mechanisms, and also the speed of DNA replication. So it takes longer to build a Mercedes than it did to build my old 1970 Ford F-150. So all else equal, the faster if you're a microbe, right, and you're in a resource-rich environment, you want to divide fast. Right, so the faster you can replicate your genome, the faster you can divide. Similarly, you don't want to spend all this energy and biological building blocks, on DNA repair. You want to turn it into offspring. And if those offspring have a few mutations, well, so be it. Okay, so basically this is what I just told you. So most evolutionary geneticists probably believe the mutation rate is determined by a trade-off between direct selection acting to tamp down the mutation rate to minimize the number of deleterious mutations, but indirect selection to minimize the resources you have to spend and the time you have to spend, you being the cell, right, to replicate your genome. Does that sound reasonable? So there is another possibility, and I call it, but mutation selection drift balance, and I will elaborate on that in the next slide. And my boss with the extra bone on his neck has coined the term the drift barrier. Remember, in infinite population size, there is no random genetic drift. The smaller the population, the more random genetic drift there is, and the more important it is. So the idea looks like this. So this is a figure from Mike Lynch and others. So the left panel is the important one here. So the idea is, there is so trait performance, that's whatever, right? But in this, you think fidelity of DNA replication. That's sort of like 1 minus mutation rate. So, and there is some arbitrary level of perfection that can be achieved. And natural selection is, sort of wants the trait performance to, natural selection wants the trait to be at perfect, right? But the smaller the population is, the more important genetic drift is, and the less efficient natural selection is. In an infinitely large population, natural selection always gets its way. In an infinitely large population, natural selection gets what it wants. Because any infinitesimal difference between two types, genotypes or alleles, the better one wins out. When the population gets really, really small, past a point, selection can be sufficiently weak to where just random sampling will outweigh very weak selection. So the point being is the larger the population, the closer to perfect a trait can get. And in this case, the trait is the mutation rate. So are there any questions? I guess feel free to interrupt me and ask questions because I kind of plan on that. You can sort of roll this around. And this is just telling you, this is where, we're looking at mutation rate. This is the drift barrier. And if you start out with a mutation rate, higher, you will go down, you will achieve the, you'll eventually get to the, the rate that natural selection, mutation selection drift balance. And if you start off with a mutation rate that's too good, right, eventually it will drift upward.
So, in other words, what you're saying is that the mutation rate is a trait in itself.
Yeah,
right?
yes,
Hair color
yes.
or skin color.
Exactly,
or height.
exactly.
So, and depending on the size of the population, you can have,
Yeah.
it might be affected more than others. So if you have a large population, then the mutation rate is going to be, is it going to be similar to all the mutation rate, to the changes in other traits? So the mutation rate, does it
So
similarly
that's
to other rates?
that's very interesting, and if I had an extra hour to talk, I would show you data on it. It kind of does.
Okay, that's what I was thinking, that maybe.
But I think that's
theoretically you would think that.
that is a insightful point, which is the mutation rate is just another trait, like size or age of maturity or femur length or Drosophila bristle number or anything, right? And therefore it itself is subject to all those evolutionary forces, right? So that's a key insight. Yeah, mutation rate is just another trait. Yes, sir.
Just to make sure I'm reading the graph right. So you're saying the drift barrier is the black line? Natural selection is the red and drift?
Yeah, here.
Okay.
Yes.
So the larger the population gets,
the larger the population, the closer you can get, the closer natural selection can bring whatever the trait is to its theoretical perfect state. All right. So, science would like to be predictive. So can we make predictions about what the optimum mutation rate should be? And that's sort of, I say yes with an asterisk, and the asterisk is all else equal. And I think on an absolute scale we can't, but we can say relatively, if we have two groups that differ in a consistent way, would we predict that the mutation rate would be higher in group A than in group B? And that's that level of predictive power I think we do have. Now, of course, our predictions might not be right, so we do experiments and observations to see. And if they're wrong, then we say, okay, well, if theory predicts this, but we consistently observe something else, then your best bet is there's something wrong with your theory, and that would be the first place to look, right? So theory predicts that asexually reproducing organisms should evolve a lower mutation rate than sexually reproducing organisms. And the reason being is that if you reproduce clonally, all of your offspring get your genotype, right? Whereas if you reproduce sexually, you're recombining with other individuals in the population. Your offspring only get half your genes, so your genes get diluted out over time, right? So if all of your offspring have your exact same genotype, then natural selection is stronger to reduce the mutation rate. And I can talk more about that later if you want. Similarly, self-fertilization, you guys know self-fertilization in plants, but also lots of animals self-fertilize. The worms I work with self-fertilize, which makes it really convenient. And all else equal, we predict that self-fertile organisms should evolve a lower mutation rate than outcrossers, provided the mutations, or the deleterious mutations, are recessive on average, or partially recessive, and there's a lot of reason to think that they are. So those are just kind of two obvious predictions. So I said, okay, so here's an intriguing example from E. coli, which I had nothing to do. This is not based on my research. These are data from a paper in Science back in 2003. Biadov is the first author. So what they're plotting is this is a frequency distribution. So they got like 700 something natural isolates of E. coli. They went out and sampled E. coli from poop from 700 different types of organisms, I guess, or so. And so this is a log scale, and this is the mutation rate. So there is about a 1, 2, 3, there's about a 5 order of magnitude, certainly 4 orders of magnitude variation in mutation rate in the sample of 700 strains of E. coli, and that's a lot of variation. And I say the intriguing example is if then you look, say, okay, what is it? Is there something special that these high mutation rate guys have relative to these low mutation rate guys? And the answer is yes. And you might think it has to do with sort of, the historical relationships, the phylogeny of the E. coli, and it doesn't. It has to do with what kind of animal they were taken from. And E. coli taken from carnivores up here have much higher mutation rates on average than E. coli taken from herbivores. And I guess, which is kind of, and being an evolutionary biologist, you get a card to tell, when you get your Ph.D. in evolutionary biology, it's like getting a journeyman's card to tell stories. Am I right about that, Matt? Totally right about it. So maybe, but remember the environment, and actually that last example has something to do with the environment. And man, I'm going way slower than I expected. Actually, no, I'm going exactly as slow as I'd expected. So organisms that live in mutagenic environments all else will need a more efficient DNA repair mechanism to achieve the same mutation rate. So for example, if you're a plant that lives on a mountaintop where there's a lot of UV radiation, UV is mutagenic, all else equal, to maintain the same mutation rate as its brother down there in the valley in the shade, it's going to have better DNA repair. Right? So if two organisms have identical DNA repair machinery, the one that inhabits the less mutagenic environment will have the lower mutation rate. And now I have the intriguing example from mice, again, having nothing to do with my own work. Jim Quinn at McMaster University in Canada, he's not really, he's an ecologist, but he did this cool experiment. We put mice, inbred line, genetically homogeneous mice, in cages downwind from the Stelco coking ovens, so a steel mill. And these steel mills, they barf out a lot of particulates, coal and steel microdust and stuff. And one treatment got a HEPA filter, so it's filtering out the particulates, and one treatment didn't. Mice were just breathing the regular old air that everybody in downwind of the steel mills in Hamilton, Ontario breathes. And the mice with the filtered air had lower mutation rates at certain classes, VNTR loci. And nobody's, I have not yet heard a good explanation for why breathing, fine particulates would increase the mutation rate. There was speculation, well, maybe there's mutagenic chemicals but still it's got, these are germline mutations. These are not somatic mutations. It could just be sort of more general stress, and that may well have something to do with it, which is one of the things I've worked on a fair bit recently. Okay, so Okay, so with 15 minutes left, this sort of gets to my research program. So understanding variation in the mutation rate. Also understanding variation in the effects of those mutations, but I will leave that. So within genomes, so are, for example, sex chromosomes more differently mutable than autosomes? Maybe, maybe not. Regions of low versus high recombination. So crossing over, you guys remember crossing over? So crossing over does not occur at random on a chromosome. There are regions of, there's recombination hotspots, and there's recombination cold spots. If you go out in nature and look, there will almost inevitably be less genetic variation in regions of low recombination. Maybe that is because recombination is mutagenic, or maybe it is something else. And there's lots of other stuff that varies across the genome, and we're interested in sort of finding out what are the sources of mutability. Within species, so my species is mostly now a C. elegans, but also other Caenorhabditis. So for example, you versus me, males have higher mutation rates than mammals have higher mutation rates than females, and and it scales with age. Jim Crow, who was a great, one of the titans of population genetics and evolutionary biology, said, the greatest genetic threat to our species is fertile old males. So I sired my poor mutation-loaded daughter when I was 46. So by all rights, she should have both eyes on the same side of her head or something, although well, it turns out most of these effects manifest themselves in behavior, and I'm starting to see that. Anyway, variation among individuals. So between species, do these selfing species of Caenorhabditis have different mutation rates, have lower mutation rates than we would expect, compared to, which is what we would expect, compared to outcrossing species of Caenorhabditis, of which there are many. And we haven't looked very closely, but the answer seems to be yes. But the data are really pretty crappy. And then various molecular properties, if two groups do differ, do they differ in every particular kind of mutation, or based, AT to CG substitutions, or is it transitions or these are different kinds of mutations, insertions or deletions or transposable elements or inversions or what have you. Do the different kinds of, if two species have different mutation rates, does everything mutate differently, or is there only some subset of possible types of mutation? So the big question then is, are there predictable differences in mutation rate? And I got 12 minutes left, so I think, actually I'll go back, I will end here and just to tell you how we do this, and then we'll throw it open to the field. So what we do what we call mutation accumulation experiments. And they're really simple. A mutation accumulation experiment is just a big pedigree. So you guys learned pedigrees, So this represents a pedigree. This is the, this is the great grandmother and father, because these worms are self-fertile hermaphrodites. So what we do is we inbreed that population until it's very, very, very genetically homogeneous. For all practical purposes, it's homozygous for the same allele at all loci. Then what we did is we made 100 subpopulations. So we let this guy reproduce. It'll have about 200 or 250 offspring. We take those offspring, put one offspring on its own plate. So now we have 100 plates, each that has one self-fertile hermaphrodite on it. And we do this for a really long time being 250 generations in this case of this experiment, which is every 1,000 days. And this is the universal PowerPoint symbol of mutation. And at some point, genomes mutate every generation. If mutation happens, it starts out as a heterozygote. It will either be lost or it will fix. And every generation, we're picking one worm. So the effective population size is 1. Remember what I said there. selection is as inefficient as it can possibly get. So all but the most highly deleterious mutations will act as if they are not subject to natural selection. So this is why worms are great for a lot of reasons, but by far the greatest reason worms are great is I can now take this population, the ancestor, I can freeze it, I can cryopreserve it, and come back, sometimes decades later, thaw it out and have live worms that presumably have not mutated in the -80 freezer or in liquid nitrogen. This works great as long as your freezer doesn't melt down. I have had freezers melt down. But anyway, so what we can do then is we can come back, and after a long time we can compare the mutation-containing ancestors to their, we hope, unmutated, or the mutation-containing descendants to the unmutated ancestor. And what we expect, mutations are bad, so we expect that fitness will decline over time. And we expect these guys are initially genetically identical, so there's no genetic variation. So as mutations accumulate, you have unique mutations in each line, so the genetic variance will increase over time. So I think I will go right to the end. And this was my lab circa about 3 or 4 years ago. I need to, if anybody is a good web designer, let me know, and I can update my website. But anyway, so now I think we'll just throw it open to the crowd, and if you guys have any questions or anything, I'm happy to entertain them.
Thank you.